Okay, this is part 2 out of 4 parts about how to reduce the risk of seizure in patients with epilepsy. So first thing, a neurologist will tell you is the patient should always take their medicines as prescribed, their anti-epilepsy medicine. And they can check a blood level to see if they're in the therapeutic range or at least close to it. And, they'll correlate that with how the patient is doing. You can also, of course, avoid strobe lights. These are things that the neurologist will tell you. Avoid sleep deprivation and stress because they're associated with increased risk of seizure. And this is where it starts getting interesting because I read avoid stress, avoid sleep deprivation. I thought to myself, well, gee, everybody knows that's increased catecholamine and increased cortisol. Everybody knows that. But how come they never talk about all this other stuff? And they just don't. I went to a whole bunch of neurology sites. And they none of them spoke about it at all. And it's just sort of like they don't know anything about nutrition. So it takes away from this whole big part of their own field. They also will recommend, avoid alcohol and avoid traumatic brain injury. These are things that are known by neurologists and are told to patients. There's other stuff that they kind of know. They just don't tend to talk about it that much. They know that, you want to avoid, be careful with fluorescent lights because they flicker. But you don't ever see them say that. But fluorescent lights can flicker and be kind of annoying, so you would want to avoid things that flicker because they're like a strobe light. TV sets can flicker. Playing video games, especially in a dark room, that has like a flicker effect that can be disconcerting to the person. You want to watch out for rebound hypoglycemia. So I wouldn't drink any really sweet beverages. Sweet beverages with no fiber can lead to rebound hypoglycemia. Your blood glucose goes up real rapidly. The pancreas overcompensates, produces too much insulin, then it comes down suddenly, and that can leave the brain kind of hypoglycemic. Not good. The other thing that I came to the conclusion is, well, once you start talking about excitotoxins, things that increase glutamate release in the brain cells or increase glutamate in the synapse, excitotoxins include MSG, monosodium glutamate, MFG, manufactured free glutamate, maybe excess sodium. There's a long story how that causes activation of the sympathetic nervous system, thus more cortisol, thus more glutamate at the synapses. So I don't know how much that's associated with it, but I would be a little careful about that. Aspartame, you get increased aspartate that can activate the NMDA receptor from aspartame, the sweetener. Glyphosate is excitotoxin. It can increase activation of SLUT through the glycine receptor on the NMDA receptor for glutamate. Caffeine causes the same thing, increased cortisol, increased catecholamines. So I would avoid it. There's a little bit of controversy about that. When you start reading the papers on the internet, whenever something is really profitable, they always try to downplay all the problems with it. And you run into weird things like you come into the papers, there won't be anything listed for, cited by or similar papers. It's almost like a lot of times there'll be no abstract. There'll be paywalls. The thing will be covered up by a twilight screen. But anyway, sweetened beverages like caffeine can cause rebound hypoglycemia that can increase your risk of seizure. And I also started thinking, well, gee, other things can have an excitotoxin effect if they inhibit mitochondrial function. So I started looking up some of those, and I found a couple of those were associated with increased risk of seizure. In particular, for example, selective serotonin reuptake inhibitors, drugs like Prozac, and other antidepressants as well. If you do a myelogram where you inject contrast, through the lumbar spine, lumbar puncture, and it drifts up to the cervical spine in the brain. We do that to look at the spine, the central canal, in patients especially who are contraindicated to MRI. By the way, I've done hundreds and hundreds of myelograms. So before you do a myelogram, we won't do a myelogram on a patient unless they've held off on any type of antidepressant for at least 2 days before the procedure, because it lowers the seizure threshold, increases the risk of the patient having a seizure, because potentially, theoretically, the contrast from the myelogram can irritate the nerves, the brain tissue. I've actually never seen a seizure from it. And the contrast that's used in the last, 25, 30 years is much safer than the stuff that was used about 40 years ago. But that's still one of the parameters in the patient pre-procedural preparation that we have to pay attention to. Okay, avoid processed foods. And I'll explain some more of this in a sec. Just think about a typical processed food. It's going to have artificial dyes in it, which I forgot to look up that for seizure, but I know that causes increased risk of hyperactivity, and I worry about that. Also can cause headaches, insomnia. I don't know if I found a paper on that for seizure. I'll double check because I looked up a whole bunch of papers. MSG MFG, we talked about that. Those are excitotoxins, brain stimulants. Glycine phosphate, increasing excitability of the NMDA receptor. Mold inhibitors are mitochondria inhibitors quite often. They're also fungal inhibitors, same thing. High fructose corn syrup routinely has HG in it. It's commonly contaminated by that. That's a major neurotoxin, mitochondria inhibitor. BPA liner, mitochondria inhibitor. That's like the plastic liner. Let's say you have a can. The can will be aluminum. The BPA will be inside of it, and it'll have gaps in it that expose you to aluminum. And it itself is not only estrogenic, it's also inhibits intermitochondrial membrane function. Some of these preservatives are rather toxic to the brain. Hexane is commonly used like processing soy. It's a neurotoxin. Atrazine is a mitochondrial inhibitor, and it's an estrogenic chemical. Sodium benzoate is also rather toxic. I'll show you a paper on that. I would avoid MJ. I would avoid sulfites because they have some association with excitotoxicity. You'd be better off having your computer hardwired to decrease EMF. If you use a cell phone, talk into the speakerphone when possible. VOCs, volatile organic compounds, can also have some risk. I'll get to a paper on that in just a little bit. Excessive sodium can activate sympathetic autonomic nervous system. I'll show you a paper on that. Some people might benefit from avoiding gluten. Now that gets a little complicated, and that's going to be less common. I'm not going to get into all that stuff with the elimination diets, but what I'm just saying, gluten was named because it's got a lot of gluten in it. But if it's not that processed, then it'll come out slow. But that's a lot better than having it manufactured free glutamate. So I don't know exactly how important that is, but if somebody was having problems, that'd be one more thing you could consider to see if you could reduce your risk of that. We talked about the bliss point, the way companies will try to get the maximum amount of MSG, MFG, sodium, high fructose corn syrup, fat to hit the bliss point where patients are most likely to become addicted to the food. Processed food is just bad. You can't win with processed food. There's so much, it's not designed to be healthy. It's designed to be hyper palatable to make you like it so much you'll be addicted to it. So we talk about how you get manufactured free glutamate. This comes from this good book, Catherine Reed, when she wrote about Fat, Stressed, and Sick, about how her kid had autism from excessive MSG, glutamate exposure, and MFG. So you just break up the individual protein, like a string, a necklace of amino acids, into their individual component amino acids, and you get all this glutamate freed up. So now it's been manufactured, and it's free glutamate. And it's the glutamate that matters, not the MSG, the glutamate. And that can overwhelm the body's ability to sequester it. Okay, here's one paper talking about MSG and epilepsy. MSG was associated with increased brain glutamate and increased risk of epilepsy. It's an excitotoxin. Okay. This one was an entire book, a research paper made into a book, monosodium glutamate effect on plasma cortisol, and it increased cortisol. Well, cortisol is going to increase glutamate, and this is a little bit of an excitotoxic vicious cycle, if you see what I'm talking about. So when you feed in, this processed food, the MSG and the MFG, that's going to increase cortisol. Well, cortisol is going to increase glutamate, and you've almost created a bit of a vicious cycle. So the smart thing is just avoid MSG and MFG, okay? And here's other things, feeding and stress, caffeine, sleep deprivation will increase cortisol, okay? This just gets a little more complicated. We're not going to go to all the detail here, but I'm just letting you know glutamate signaling is more complicated than just AMPA and NMDA. For our purposes, that's all you really need to know. AMPA is a sodium channel, NMDA receptor is a calcium channel, but it does affect other channels. One of them is the metabotropic glutamate receptors. So these are called ionotropic because they let ions come through, like a current. Whereas this is metabotropic, like through metabolism, it activates a pathway. Okay, and the pathway then releases IP3 from the inner leaflet of the plasma membrane, and that activates opening of calcium channels, IP3 channels on the endoplasmic reticulum, and lets more calcium into the cytoplasm. And it can even let more calcium come in by secondarily opening up these what are called ryanodine calcium channels on the endoplasmic reticulum. Endoplasmic reticulum is a calcium storage site. And a certain amount of this is useful. It's all part of long-term potentiation for learning and memory, okay? So there's a good reason to have all this calcium stuff. You just don't want it activated unless there's a good reason for it. You also have an additional calcium channel called the TRPC3, transient receptor potential canonical calcium channels, okay? That's why it's C3. And these are also related to learning, BDNF activation, localized synaptogenesis, neurogenesis. So these are all things that a neuron's normally supposed to do. What happens, though, if you overactivate this guy, NMDA channel, you get too much calcium coming into the cell. All right, I'm going to show you all this, and later on I'll show you one more calcium channel, but the important point is, in order to get calcium out of the cytoplasm to turn the cell off so it doesn't overwork itself, go into excitotoxicity, you need ATP. And the reason you need ATP is to run all these pumps. So that is an investment of energy. Because whenever you pump calcium out of the cytoplasm, you're always pumping it against the concentration gradient. The concentration of calcium outside the cell is about 15,000. That's a lot. 15,000 times higher than it is inside the cell. So to pump it out there, you need to use these ATPs. This is related to the potassium-sodium pump, and then secondarily, that is used to create energy for the knockout exchanger, sodium-calcium exchanger. So the bottom line is you need ATP to pump calcium into the extracellular matrix. You also need ATP for the circle pump, the endoplasmic reticulum calcium ATPase, to pump calcium into the endoplasmic reticulum because it has a very concentrated storage of calcium inside of itself. The S just stands for sarcoplasm, and that comes from muscle, but in a brain cell, really only endoplasmic reticulum is relevant, but that's how it gets its name, circa. Sarcoplasm, endoplasmic reticulum, calcium ATPase. You also have a plasma membrane calcium ATPase over here, which is also ATP dependent, but it's relatively slow. Knockout exchanger is much faster. And another thing you'll notice is you need magnesium to run all these ATPs, ATP-related enzymes and pumps. So if you're magnesium deficient, you're kind of screwed because you need magnesium. It hangs on to the second and third phosphate so that this reaction goes correctly. Its positive charge helps to control the negative charges of the phosphate so they don't pop away from each other faster than they should. You'd also ideally have the proper dietary balance of potassium intake from plants and sodium intake. Normally we should be at least like 10 to 1, 20 to 1. With more potassium than sodium. But in the modern world, people eat all this processed food, and they're often 10 or 15 to 1 times more sodium intake than potassium intake, which is bad. That can dissipate these plasma membrane gradients and decrease cellular function. So eating the plant foods gives you the best of everything. The more potassium, less sodium, more magnesium. That's what leads to healthy functioning cells. Okay, and then you need that ATP production coming from your inner mitochondrial membrane. And I've said this before, but when I went through I knew mitochondrial inhibitors had to be important. But when I went through, I own like about 7 biochemistry books. Not a single one of them had anything useful on mitochondrial inhibitors. I'm like, how could that be? So I started going through the literature, and I quickly found over 50 mitochondrial inhibitors. And that's highly relevant because you need this inner mitochondrial membrane to produce ATP. That's where the vast majority of ATP in the human body is produced. And so when you inhibit this, you're not going to be able to make as much ATP. ATP is adenosine triphosphate, which is like having a $20 bill in the cell for getting work done. And because you can't make all this ATP, then you're going to have a hard time pumping calcium out of your cytoplasm, and that's going to lead to calcium accumulation in the cytoplasm. And calcium accumulation in the cytoplasm, that's going to lead to excitotoxicity, overactivation of the cell. And you'll notice that a lot of these things are really common. Glycine phosphate, glyphosate, okay, inhibits complex 2. Antifungals, mold inhibitors, inhibits complex 2. Metformin inhibits complex 1. Now, they say metformin and statins don't increase the risk of seizures, and so maybe they don't. But I always worry about the papers written on popular things. Like these are very, very popular drugs because there's so much industry pressure to make the drug look good that you don't know if you're getting reliable information. So there's lots of common things. Cadmium is involved here, F-, H&E, hydroxynonenal from omega-6 cooking oils. There's atrazine, there's BPA. All these things are inhibiting your inner mitochondrial membrane, your electron transport in your mitochondrial membrane. And there's other real common things, fluoroquinolones, beta-lactam penicillins, aminoglycosides, tetracyclines, beta blockers. A lot of food dyes will do it too. So there's a good mechanistic reason why these might cause excitotoxicity, and in so doing, causing excitotoxicity, they might also increase the risk of having a recurrent seizure. That's why I mentioned them all. Okay, so anyways, that's I thought that was kind of interesting, and that's sort of, I think, was sort of my contribution, is that I know a lot about what causes increased glutamate release, what has an excitotoxic function, what has a secondary excitotoxic effect, and all of these things will be likely to increase the risk of seizure. So I look for more papers. I'll show you in part 3. So this is the conclusion of part 2. In part 3, I'll show you more of the papers on the things increasing the seizure risk. And there's a big deal for a young person newly diagnosed with epilepsy. If they can reduce the number of these things as much as possible with diet and lifestyle, they can reduce the number of medicines they need to take and their likelihood of a repeat seizure, and that will improve their quality of life. So anyways, we'll start with part 3 in just a moment here.